Good evening all of you. In today's botany session, we are with the chapter Principles of Inheritance and Variation, but which topic under that? Linkage and Recombination. To get into this topic, let me say you some basic points for the student, those who are listening this chapter or the related thing to that first time. What are that basic things? Like if I am taking the term chromosomes, right? Now there, can I say this is a chromosome, right? This chromosome will carry many genes on that. What I am talking about? Genes. What are that genes which are controlling our characters, means our phenotype, what we are looking from outside, or any other organisms, whatever they are expressing from outside, that will be controlled by the genes, and these are aligned on the chromosomes, right? Now these genes will be traveling with this chromosome from one generation to next generation. Maybe all as such, maybe after crossing over. Crossing over means if I am taking the 2n number in human beings 46, 2n means total number of chromosome 46 in humans. Suppose I am taking that. Maybe you have heard that it is making 23 pairs. Means every chromosome have its homologous. So rest is another story, that during the S phase they make the two chromatids on the same chromosome, that is a separate story. But right now I am focusing that genes, suppose I am taking A, B, C, D, E, F and G. Suppose this is for height. On the same position on its homologous chromosome, they will have allele or a gene for a height. Can I say this pair you call it as which type of chromosome? Homologous chromosome, right? So if it is showing the crossing over means this section coming on this side, this is going on this, means a segment of DNA can exchange between the homologous chromosome, right? I am not considering this thing right now, simply I am saying that genes are present on the chromosome, right? Now during the meiosis, what happened? Meiosis is a reductional division which made 2n number as n. Means like in humans, when gametes are forming, 46 chromosome will remain 23 chromosomes. That is the reducing number. Meiosis do it like that to form the gametes, so that when the gamete fuses male and female, again we can get back 46 in the new generation, right? So in the meiosis, what will happen during the meiosis in the gamete formation, especially in case of human beings? Can I say this chromosome will go on this side and this chromosome goes on this side? Why I say usually in case of human beings? Because in plant the gametes are mostly formed by mitosis, and in animals the gametes are formed by the meiosis, right? That's why I spoke that term. Okay, let's make it more simple. Let's recapitulate the things to make it simple. We are having chromosomes, and on the chromosome what have been aligned? Genes. And every chromosome have its, you can say, the same copy, right? So can I say, suppose my hand is there thinking like that, 5 chromosomes and 5 chromosomes same like that, then it is 2n number, right? If I am taking this 5 separate, this 5 separate, n haploid, right? That will be achieved by meiosis. So means the chromosome which are homologous are going to separate. During which process? Meiosis, right? Number one, you should know about this thing. Very important, I am repeating again and again, where the genes are located? On the chromosome. Can I say the genes which are present on this chromosome will go along with this chromosome, and genes which are present on this chromosome will go along with this chromosome. It is simply like what thing? Think about two buses are there, bus A and bus B. In bus A, the passengers which are sitting will obviously go where the bus A will go, and the passenger in the bus B will go where the bus B will go. And keep in your mind that two buses are not dependent upon each other. They can segregate independently. But you are having two categories of passengers. What are the two categories? One which is present in a same bus. That you call it as what? Dependent passengers. They have to go in one direction. But the passengers which are present in B bus, that will be independent from the passengers of bus A. So can I say again, two types of passengers? One which are in a same bus, either of A bus or B bus, all depends upon each other. They have to go in one direction. But the passengers of bus A and B, they are independent. So buses are always independent, but the passengers what are sitting there, they are dependent upon the bus where it is moving. So what thing I am relating? The buses are equivalent to what thing? They are equivalent to chromosome. So where the chromosome will go, their genes are going in that cell or that progeny. Are you getting that? So keep in your mind the two types of gene right now I am discussing. One is which are dependent, which are present on same chromosome. Can I call that dependent genes as what? Linked genes. And which are independent on different chromosome, you call it as unlinked genes, right? So I am coming back. What is our today's topic? Let me to introduce back again. Our topic is about linkage and recombination. To understand what is the linkage, you should know we are reading the word linkage in which context? Related to the genes which are present on the same chromosome. So those are on different chromosome, although they are homologous. They are present on different chromosomes. Then they will be what? They are not going to be linked. Linked genes are what? Which are present on the same chromosome, right? Now that linkage we have to discuss. Means I am talking about genes which are present on the same chromosome. This is our topic, and how much recombination they will show, and who have proved that thing experimentally, by which organism, and how he discovered all the thing experimentally, we will discuss in our today's session. Fine. So here I repeat, chromosomes, genes present on that, where the chromosome will go, their genes will go in that direction. For this chromosome, these all genes are linked. But how much they are linked, that all depends upon one relation. Let me to say you that relation. You must learn this formula if you are not getting even the lecture at all, right? Which I am assuming it is not possible. If you are paying your attention and I am giving my best, and hope so you will get overall the concept of this topic. Fine. So listen this point carefully. This will help you to overcome the lacuna of the you can say understanding of the concept. What I want to say, linkage, how it is related to the other things like recombination, which we call it as crossing over, distance between the genes, that all. So linkage is inversely proportional between what thing? Distance between the genes, right? Distance between the genes. Which genes I am talking about? In full topic I will discuss about the genes which are present on the same chromosome. So here the linkage on the same chromosome of the genes, how much they are closely linked, that depends upon the distance between the genes. Means it is inversely proportional to that. And even it is inversely proportional to crossing over. Just now I said you what is crossing over? Exchange of the segments between the homologous chromosome. So that exchange will be how much possible? That depends upon the distance and how much they are linked to each other. I will again introduce all these things by taking the example of some chromosome. So here linkage is inversely proportional to the distance between the genes. Even it is inversely proportional to crossing over, and even it is going to inversely proportional to the non-parental types. Means new combinations are going to be formed, right? So this is the relation among these terminologies. Now let's talk about the great scientist who actually introduced the term linkage and recombination. Let me to say what is a recombination here right now. This crossing over, you can call it as gene recombination. Fine. So if I am asking you how much linkage and recombination, how they are interrelated, they are inversely proportional to each other. Fine. Now who introduced the term linkage and recombination? That was the scientist Thomas Hunt Morgan. He introduced these terms, and what he said, what is a linkage? Can I say this is the physical association of the genes, physically where they are present. If they are away from each other, like for example in this chromosome, can I say A gene is very much far away from gene G? The distance increases. Can I say the linkage, right? That is going to be decrease. For example, think about a family is traveling in a bus. On one seat parents are sitting, and on one seat their siblings, means their kids are sitting, means siblings are sitting, and that siblings start fighting. They will increase the distance, so obviously one sibling will be adjusting with the mother, one is with the father. Can I say if linkage is going to be less, right, then can I say distance increases? That will create the new recombinations. Here is what is the new combinations? That will be the combination not with the sibling with sibling. It is now with the mother with one sibling and father with the another sibling. In the same way here, once the distance is more, then the crossing over chances will increase. Means this A can be separated from G during the crossing over, because this A will come on this side, small a will come on this side, right? During crossing over, during which process? Meiosis. You will learn in chapter cell cycle, cell division, under meiosis 1, under prophase 1, under packaging, this exchange can happen, but between homologous chromosome. So if the distance of the gene increases, then that is more chances that the gene can going to be exchanged by the homologous chromosome during the crossing over. Now can I say A and B are more close? When the closeness is more, can I say linkage is more strong? If linkage is more strong, can I say distance is less? If distance is less, can I say chances of crossing over will be less? Then what will happen? AB can go as such on this, and small ab can come here together. So the chances of capital A, capital B to go together on other homologous, means on this homologous chromosome, is going to be more, together will be more. But now AB coming on this side, can I say it will be now separated from G? Why it is separated from G? Because the distance between that is much more. Let me to draw that scene what I am telling you here right now. Like suppose I am taking the same condition in this way. Now this chromosome, if I exchange the genetic material after crossing over, AB will come here, here will be capital C, capital D, capital E, capital F with capital G. What will be on this chromosome? Capital A, capital B. Now here will be small c, small d, and small e, small f, and small g. So can you see here capital A, capital B are more close, that's why they are going together after the crossing over. During gamete formation, I can say during the meiosis. So AB is now separated from G or F or E or D or C. Reason why? Because their distance is going to be more as compared to AB distance, right? That's why we say here, if the linkage is going to be more, the distance will be less, and obviously crossing over chances will be less, and the non-parental types are going to be less if the linkage is going to be more, and vice versa of that thing. Coming back to that scientist, what scientist I discussed with you? Thomas Hunt Morgan, who had given this concept of linkage and recombination, and he put his work on the organism, you call it as Drosophila. The complete word is melanogaster. Common name you can call it as fruit fly as well. Now, why he had chosen this as a specimen for his research, especially for dihybrid crosses which are linked to the sex chromosome. What are that sex chromosome? That are allosomes. It can be XX like in Drosophila for females and XY for the males. Now, Thomas and Morgan, why he selected the Drosophila melanogaster? Because it is easy to culture this organism on the synthetic medium. What we are giving, they can easily grow over there in the synthetic medium. Second thing, this organism is having the life cycle of hardly 2 weeks, very short, and in one mating it can give many progenies. Many young ones can be there in one mating, because if you have much more example to show your result, it will be more authentic. If we are showing it with one organism, it won't be that much authentic. We cannot rely upon this thing. That's why many progenies make us to rely upon our results. Like that Thomas Hunt Morgan did with the results of the Drosophila melanogaster. Even this organism, they can show the sexual dimorphism, or we can identify males and the females. When you will see your NCERT book, you will find they have shown the picture there for male and female Drosophila, and you will find female will be larger in size and male will be shorter in size. And even it can show chromosomal dimorphism, means XX is representing what thing? Female, and XY is representing the male over there. Fine. So, some of the points I have written, that why Thomas Hunt Morgan has used this organism for its experiments, and due to all this experimental genetics he had performed to prove his result, that's why you call this scientist as father of, you call it as father of experimental genetics. Right? So, you should know about this small information to start with the process of linkage and recombination. So, to prove this, some examples of linkage and recombination, we have to pick up the Morgan's dihybrid cross. Now, let us see what do you mean by the dihybrid cross. Dihybrid cross means when we are selecting two characters and studying that how it is going to transmit from generation to generation. When their hybrids will be forming, definitely it will showing the two characters. Now, which characters Morgan has selected, we will pick according to your NCERT book, right? So, for that thing, I want to show you one tabular form to clear all these things. Number one, you should know the genes, I already said you present on chromosome, right? But these character, what now I am going to explain you, will be present on which chromosome? That will be present on X chromosome. That's why you call it as sex linked characters, being present on sex chromosomes. If I say for Drosophila, how many chromosomes it is having? 2 and number 8. Can I call it as 4 pairs? Out of 4 pairs, 3 pairs will be of autosomes and one pair will be for allosome. What is that? You call it as sex chromosomes, which will determine whether it is a male or the female Drosophila. XX is indicating for the female, XY is indicating for male Drosophila. So we will discuss in the linkage and recombination part the genes which are present on XX chromosome, or you can say on the X chromosome. What genes are present on X will not be present on Y. We will take Y chromosome just to say that it is a male Drosophila. Whatever is now on X, it is going to express in case of the male. Now coming to the same slide back again. If I am taking now, these are the characters. Every character will be having two traits. Right now body color is a character. What are the traits for that? Brown and yellow. Maybe in your junior classes you have learned for the pea plant, the Mendel has kept his work. So he had chosen like character stem height. For example, what are its traits for that? Tall and dwarf, right? That will be the traits if you go literally with the terms. Now here what is that? For Margon experiment, he followed the Mendelism only, but for the linkage we are not getting the Mendelism. Then the ratio what you will get from monohybrid or dihybrid, now we are discussing dihybrid, the ratio should be 9:3:3:1, right? So if you are not aware, you are newcomer for this topic, don't worry, simply just right now you should know these are the characters which an organism is showing from outside. So every character has two traits, like a plant can be a tall, if tallness is not there, what is opposite to that? It will be dwarf. Same like that, this character body color is having two traits, one is a brown and one is yellow. But out of these two, which is dominant? Dominant means who will be expressed when it is present. Either one allele or both the alleles on both the chromosome, if dominant, then definitely it will express either one or two, definitely it will express. But recessive should be on both the chromosome. What both chromosome I am talking about? Homologous chromosome, right? I already mentioned you this point. The chromosome, if I am taking like this, right? I hope so this is visible. This is a chromosome, this is a chromosome, homologous chromosome. If I am taking the body color brown and yellow, right, then this will be showing, suppose body color, it can have Y plus, Y, or it can have what thing instead of this thing? It can have instead of this thing here Y and Y. If both on homologous chromosome they are having the recessive alleles, then it will take what type of trait? Recessive trait. And if one is dominant at least, then obviously which trait will be expressing? Dominant trait. Now let's see how they put the indication, how we have to mark the genotype and the phenotype in the Margon's experiment. Now here the dominant trait is brown and recessive is yellow for body color. Eye color, what is that? Red color is dominant, white is recessive, wing size normal is dominant, miniature is a recessive. Now how to express in a genotype? Genotype means actually on the genes, whether it is a dominant or recessive allele, that is a genotype. Phenotype means outside, how you can visualize an organism. Outside you will see whether it is a yellow colored Drosophila or it is a brown colored Drosophila. But inside, due to what thing, it will be brown. It can be due to the Y plus chromosome, or it can be due to Y plus or Y chromosome, right? So either it can be homozygous or it can be heterozygous. If we need to write its genotype, then how you should write for these traits? Now see here, this is important. If you understand how to write all these things, only then you can understand the dihybrid cross of the given by the Margon, right? So brown color is expressed by white, means expressed by Y, which is the genotype we are taking from recessive trait. It is not like the Mendelism. In Mendelism, you are taking, if tall is dominant, you have to take capital T from the dominant trait first letter, and the recessive you have to mark with the small t. But here is not like that. In the Margon you have to take the recessive trait and Y should be small here, and to represent its dominant you have to write Y plus. And for the eye color, what is the recessive? White, so we have to go with small w, and to show the dominant we have to write W plus. And to write for wing size, what is the recessive? Miniature, M we have to show it, and for the normal we have to write M plus. Means where you are writing plus, that is representing dominant trait. Where you are not representing plus, that means a recessive trait. Every character will have two traits. Now in the first cross of the Margon, he had taken these two characters, and in the cross B, second cross given in your NCERT book, he had taken these two characters, right? So for understanding of that crosses, this table form is very important for you, right? Okay, then after this, if I take the concept of linkage and recombination, and that recombination is further telling, means how much percentage the new combination non-parental are going to be formed. If suppose for example, I am getting in a population of Drosophila the new combinations of the character, suppose 10%, that means the distance between that genes, like suppose I am taking A and B characters, and the distance between that gene will be how much? That will be 10 centimorgans. Simply what recombination percentage they had given you, just convert in centimorgans, or you have to convert in MAP units. Actually we are using MAP units in case of prokaryotes to say the distance between the genes, and centimorgans we are using in case of eukaryotes. Usually the trend is like that. And centimorgan, C should be small, M should be capital, because it is M capital means they are representing the name of the scientist, so M must be capital here, right? So can I say 1% of recombination is equal to the 1 centimorgan, the distance between the gene? If the probability between two genes to make new recombination, what percentage probability is there, that is actually telling you how much distance between the genes are there. Margon cannot see the genes on the chromosome. Margon is only looking to how much that character combination is coming in the population of Drosophila. If recombinant percentage is more, and I said you it is inversely proportional to linkage. If recombination percentage is more, can I say linkage will be less and distance between the genes will be more. Do remember that first formula just now I discussed with you, right? So linkage is inversely proportional to the distance between the genes on the same chromosome I am talking about, and it is linkage is even inversely proportional to the recombination, even to the, you can say crossing over, or even to the non-parental, you can say types, right? Okay, now coming back, if I am taking linkage and recombination, the percentage of recombination what I am getting, exactly that will be the value, but units for the distance will be centimorgans, or it can be MAP units. Now let's talk about this table form, recapitulate that point, because now I am entering into that two crosses, right? So have a look there, body color, eye color in cross A, whereas eye color, wing size in a cross B. And what is the dominant trait for the body color? Brown, here will be recessive yellow. Eye color, red dominant, and white will be recessive. Wing size, normal is a dominant and miniature will be recessive. And see the presentation, how you can write it in the genotype form, right? Okay, now to get into that dihybrid cross, suppose I am taking this X chromosome and this as an X chromosome. If it is having Y and W genes of the two characters, body color and the eye color. Second will even have Y and W. Overall what it will be? I can say both are recessive. I will be read at Y and W. How you will write its phenotype? You will write it phenotype as yellow white. Means it will be having yellow body color, white eye color. Fine. Now if I am taking, suppose now other thing, I am not talking about the cross right now. If I am taking here like Y, W, and here I am taking Y plus, W plus. Who will be dominant here? Y plus and W plus will be dominant. Fine. Now if dominant, can I say it will be like this thing with you? Both are dominant, so need not to write that this is brown and this is a red color. No, you simply write this is wild type. Wild means dominant for both the characters. So you should know some conventions before we are getting into the dihybrid cross given by Margon. The table form just now I represent number 1. Number 2, this how to write their phenotypes. Now if I am writing one like Y plus W and second is YW. Can I say this will be dominant? I can take it Y plus. What will be WW? It will be like this. Dominant means what? All those small y is there, plus will be dominant on this. Means which body color will be coming here? It will be brown. It will not be yellow. Now here both are recessive. Obviously which body color will be, which eye color will be there? White. So what phenotype you should write? Maybe you are thinking the body color brown and here you are thinking I should write for white. No, here simply you write the white. What do you mean by white? Writing white as a phenotype, that indicating that it is recessive for eye color but it is dominant for body color, right? Now if I am writing like Y now and here I am writing W plus and W. So can I say overall this effect will be recessive and overall this effect will be dominant. Now what should I write here? Its phenotype, we have to write it yellow. Means you have to write the phenotype based on the recessive trait. So you have to write yellow. Yellow type of Drosophila in this dihybrid cross where I am taking two characters, the body color and the eye color. If I am only writing recessive trait, that means it is recessive for that character and automatically second character will be dominant. Please do focus on these terms. If both are in recessive, then you have to write the complete term for phenotype yellow and white. If both are plus plus, means both characters are dominant genotype they are having, then you have to simply write wild. And if you are having one dominant, one recessive, then you have to give the phenotype based on the recessive. That is understood, the second character will be dominant. And if you are having same on this situation, you have to write yellow because here body color is recessive, eye color will be dominant. And here when Margon will start its experiment, this is usually the parental type because this feature they have taken in the parents of Drosophila. I will open that chart in front of you, then we will discuss. And this we have taken like these are the recombinants, right? This we are taking as a recombinants. Same you have to do with the other character. What is the other characters? I am taking now the combination of what thing? I am taking the combination of the eye color with what thing? With the wing size. I am taking like miniature. If you remember, both are recessive. And now if I am expressing on XX chromosome, from the beginning I am drawing on XX. Why? Because both the X's will have the same genes, right? And if I want to draw for the male, then I must write this as an X, then this is for Y. And I have to write the genotype only on one. Like here if I write, it will be WM. I need not to write for the males on both the chromosome because Y is not having the same genes which are present on X. But in XX female you have to write on both. If suppose it is like this, what phenotype you will write in cross B for such combination? Overall, can I say it is recessive? It will be W and M. What you have to write for this? Which eye color? White. What type of? Wing miniature. Fine. And if I am having like this, it will be W, W plus, and here if I am having M, M plus. Overall impact will be dominant, and here overall impact will be dominant. Can I say simply like plus plus, or do write it W plus or M plus? If both are plus, what should I write now? I must write it as wild, right? Because both are dominant. So what are these type? I already said in the Margon it is an parental type. Now if I am writing the combination like W plus M, and here I am writing W M, right? Then what will be there? Can I say it is dominant for one character and recessive for another? What is that? W M. Simply I am doing the suppositions. For that you can understand what phenotypes you have to write for the given genotypes. And keep in your mind, if XX, then obviously it will be a female. XY obviously the male. So coming back to this, what you are going to write? I repeat, sorry, here I must write for plus, right? So can I say one is dominant, one is what? Recessive. What should I write for eye color? It should be red, and this is for the, you can say, miniature. So I must write what thing over here? I must write for the recessive. What is that? Miniature. Means I need not to write for dominant. I have to write in such situation when one is dominant, one is recessive. Only for recessive I need to write. Now again for next one more thing, if I am writing W, W, and here I am writing M plus M. Can I say this is now giving the recessive? This is giving what thing? Dominant. So I must write which phenotype here? What is that? Eye color? That will be white. I need not to write white and the normal wing or the large wing. I need not to write. I have to write the phenotype based on the recessive. Fine. So if I am writing like this, 1 plus and 1 recessive, or 1 dominant, 1 recessive, then what type it is? Recombinant type. Fine. So please keep in your mind all these conventions in your mind before entering into the dihybrid cross given by the Margon. Because if I will discuss with you the crosses, I can say this is the end of the topic. But to build up to that important thing, you should know the conventions, what we are assuming to read it given by the Margon. Are you getting that? So please be careful about these assumptions, right? Now coming to the crosses now. So this is a cross A given according to your NCERT book. Fine. So in the NCERT book, in cross A, they have taken which characters? I already said you. They have taken the character of the body color and of the eye color. And I hope so you remember body color, what is the dominant trait over there? Brown. This is dominant, and recessive is what thing? Recessive will be yellow. And what for the eye color? It will be red dominant and recessive will be white. Please don't forget this point which we have already discussed, right? Okay, now coming where these characters are present, where these all characters are present, what we are discussing in the crosses, these are present on the allosomes, means on the sex chromosomes, right? Now here XX is indicating what thing? Female. XY is indicating what? Male. So the parental, what they have taken, YW means recessive for both the XX chromosome. If both are recessive, what will be the phenotype? I should call it as white yellow as per the rules just now I explained you. Fine. So be strong in listening from the beginning till last of the topic. Only then you can understand linkage and recombination. What I taught you from the beginning of lecture till last, all are linked with each other. If you will miss in understanding any of the points or miss to listen any of the point, you cannot understand this full course, right? So here I repeat, XX female, XY male. We are taking female, recessive for both the characters yellow and white, and male we have to take both characters as a dominant. What is dominant? Y plus and W plus, right? And you call it as a wild type. Now once they are making the gametes, this is separate, this is separate. Now can I say when this X, which is having small y, small w, is making combination with X of the male, then what type is going to be formed? Female, which is having what type? Y and Y plus, W, W plus, plus and plus will be wild type. Now if this X or this X is combining with Y, can I say we get the male progeny? But both are what thing here? Small y and small w. They are not having plus. Can I say it is having phenotype yellow and white? Yellow for body color, white for the eye color. Are you getting? Now can you see in F1 generation, there is no new recombinant. What are recombinant? One should be plus, one should be recessive. So you will not find any of such combination in F1. But one thing is important to notice here, what is the character of the male parent? It is coming in the female progeny of F1. And what is there? The phenotype of the female parent, that is coming in the male progeny of the F1. This is very important feature you should notice from the parent to F1. How the things are, or you can say the characters are going to be inherited from parent to the F1 generation. Fine. Now coming to the important thing which is going to conclude the Margon experiment regarding linkage and recombination, that is about the F2 generation. For the F2 generation, he had taken the F1 generation like as what thing? I must take it on this side. F1 generation, they have taken as a parent for F2 now, right? But actual parents are what? From where we have started. Keep that in your mind, right? So if I am taking this thing right now, focus only on that point. Don't see here and there, only focus here. F1 we got it like this. What is the female? This one, which is wild type. Male is what? Recessive, yellow and white. Please focus everyone on this point. This is very, very important conclusion now of the full topic now. So what is there? If I am taking that this XX is not showing crossing over during meiosis, during gamete formation, if no crossing over is happening, then what is the chance of parental type? Same, this thing is carried from generation to generation. This will be 98.7%. I repeat, if you cannot read from the board, it is 98.7% parental type. Fine. How it comes? See here, if this and this, it is Y plus and W plus. Can I call it as wild type of F1 generation? And here for F1, male is Y and W. What you call it as yellow and white phenotype. Such type of Drosophila you will find in nature for F1 generation for the males. Now suppose if this X with this X, can I say this female you will get? Now focus only right now in this column. Fine. So can I say this X with this X will give you this female. What is that female? Both are recessive. Can I call it as yellow white female? And if this X with this X, can I say now I will get this female? What type of female now it will be? Y plus W plus. Can I call it as wild type? Now if I am taking right now this with this Y now, if this is combining with this Y, male or female? Obviously male. because it is having the Y chromosome. So we will get F2 progeny like this. Small y, small w means yellow and white. Now if I am combining this with this now, can I say again we get the male? It will be Y plus W plus, what I call it as wild type. Now can you see here, we are getting males and the females 50%, 50%. But all the phenotypes, just now what I spoke, that all phenotypes are same like the parents ultimately. What are the parent? Yellow, white or wild. Same I am reading here. That you call it as parental type, which is same like the parents from where we have started. Then how much percentage it is there in the progeny of F2 of the Drosophila? That is 98.5%. Now what about this column? This is actually the main thing which help us a lot in arranging the genes on the chromosome, how it comes. Now see here, if I am taking the concept regarding the second column, now please focus, if this second column I need to discuss, out of 100, how much percentage this column will show, which is representing the recombinant types, that is 1.3%. When you will get this 1.3%, when the probability between the genes to get cross is 1.3%. Now suppose XX show crossing over. When it show crossing over, suppose with chance is hardly 1.3%, then what will happen? This Y plus will come on this side and this will become W. And can I say opposite, what will be coming? Y, and here will be W plus. If crossing over happen in XX chromosome of F1 generation, then you will get such type of combination. Now see how, if I am taking, now don't consider this part. For this column, you have to consider this thing, right? Now if I am saying, I am getting the combination of this X with this X, then can I say I will get this type of combination? Female will be there. What is this combination? Let me to read it for you. It will be coming out by Y and W plus. What is Y and W plus phenotype? That will be yellow. Can I call it as recombinant? Just now before that I taught you. One is plus and one is recessive, that is a recombinant. And this is a recombinant in females. And after crossing over, if I am taking next combination, like if I am taking this X with this X of the male, then this will create what type of female? White. White means it will be recessive for which character? Eye color, and dominant for the body color. Likewise, same you have to do with the X's with the Y. You will get the males with the recombinants, white and yellow. How much percentage of such type will be there in the F2 generation? That will be hardly 1.3%. If you see, parental type is much more as compared to recombinant. That means if parental type more, we are not getting non-parental more. Linkage is more between that. As per the formula, what I spoke to you in the beginning, I said you the linkage is going to be inversely proportional to the non-parental type. Now if we are getting the recombination percentage 1.3%, very less, can I say linkage will be much more? Linkage means the distance is less, they are much close, there is less chance to get separate. Separation if less, can I say recombination will be less? So what we can conclude from this cross, can I say we conclude from this cross that on this XX chromosome, the distance between Y and W genes on this chromosome will be 1.3 map units, or if it is a eukaryotes, then I must say it is centimorgans, right? Just percentage I said you have to convert into distance. 1% is equal to 1 map units. You have to calculate the distance based on percentage of recombinations, not based on the parental types. So what is the distance between that genes? That is what thing? 1.3 centimorgans according to cross A. Who had given this relation now? Linkage and recombination, fine, that was explained by the Margon. But which scientist now correlated this frequency of recombination and arrangement of that genes on the chromosome? That was given by the scientist Alfred Stroudpent. And this scientist was the student of Margon. This scientist was the student of Margon. He had given this relation. Recombinant formation, recombination is happening, linkage, the relation between that, what I have shown you, it is inversely proportional. That was mentioned by the Margon. But how it can be helping in gene mapping? What do you mean by gene mapping? Looking to the recombination percentage, you are finding out the distance between the gene on the same chromosome. That now correlation was given by this scientist, right? Now coming to the last, dihybrid cross. Why I am calling this even dihybrid? Because two characters again here. Which two characters we have taken here? Let me to write here. Can I say the first character indicating by W? Obviously it is a eye color. What is the dominant trait here? That will be red. What will be the recessive trait here? That will be white. How you are indicating it? W, this will be W plus. Second character, can I say wing size? Right, it is having what thing? Dominant and recessive. What is the dominant over here? Normal, or you can say large wing. Recessive is what? Miniature, right, which you can indicate it by M. This you are indicating by M plus. So which two characters in cross B we are taking? Eye color and the wing size. But they are present on same X chromosome. But for an experiment, we are studying on that two characters. Now we are ignoring other characters or other genes on that chromosome. So let's see the same thing we are doing like what I discussed with you earlier for the cross A. Now can you see the two genes for the female parent will be small w, small m, means white and miniature will be the phenotype. And we are crossing with such male which is having dominance for both the characters. Can I call it as wild type? Now these phenotypes are the parental phenotypes. If you will see F1 generation, in F1 what is happening? What is this generation? F1 generation, what is this thing? Parents or parental type. This X, female, this X, this Y, that will be male. So what type of F1 progeny? Wild. It will be having wild and it is having white and miniature. Can I say the female parent is coming in male progeny? Means their characters of that genes, and the characters of male progeny is coming in the female progeny. That is happening as a main question in the F1 generation. Now let's see F2 generation. In F2, if we are taking F1 is becoming as a parent for F2, but ultimate parent will be this only from where we are starting, right? Now focus only on this point please everyone. If I am taking this as a wild type for the F1 for the female, what will be for the male now? It will be white and miniature. If there is no crossing over between XX, you will get F2 like this. Females, wild type, white and miniature. Males, wild type, white and miniature. Can I say there is no change in their phenotypes? No change. It will be same like parental types. How much percentage? 62.8%. How much percentage? 62.8%, which is much less as compared to first cross. What is the first cross before that? This one. This is how much percentage of parent? 98.7%. Now if in the second cross the parental is going to be decrease, what is going to be increased now? Recombinant percentage as compared to cross A. Don't compare this two now. No, you have to compare the recombination of this cross with the first cross. Means cross A. Cross A, how much recombinant percentage we got? 1.3%. Here, how much recombinant percentage we will get? 37.2. How we are getting that? I already discussed. If suppose the crossing over happens, then it will become W plus M and it will become WM plus. Now you have to consider this for the recombination, means this will make the gametes and combine with the male chromosome and you will get a recombinant type of phenotypes which is not there in the parents. In cross B, how much that percentage is? 37.2% and present on same X chromosome. So can I say what is the position of the character wing size on here, on this chromosome, X chromosome? Can I say it will be somewhere like this? I should mention it should be like this, miniature, means I must say this is the wing size. Now according to recombination, the distance between these two genes will be given by the percentage of recombination. How much percentage of recombination? Just now we have seen 37 point, it is going to be 37.2%. See here, 37.2%. So what is the distance? Now we have to conclude from this, not percentage, now you can say 37.2 centimorgans. Now if the distance is more, that's why their recombinant percentage is more as compared to parental types. If recombination percentage is more, can I say distance is more? But linkage will be less, because linkage is inversely proportional to the distance as well as recombination percentage, or you can say number of recombinants, right? So keep that in your mind. Which gene now? See the conclusion, how we can conclude. On the same chromosome, we are having YWM on X chromosome like this. Now will you please answer me that which gene has more recombination percentage? Which gene can have more recombination percentage? Maybe you are thinking WNM because it is having more distance. But no, the answer will be YNM. Why? Because 1.3, 37.2, if you will add, it will be 38.5 centimorgans now. Can I say this is much more distance than 37.2? It is more distance than that. So if distance is more, linkage is less and the recombination chances will be more. Are you getting that? Likewise, Margon, with the help of his student, right, Alfred, from the percentage of the Drosophila in the population, they predict the position of the genes on the chromosome, how they are arranged. Are you getting that? If the percentage is that much will be the distance between the genes, right? Okay, this is a one aspect on the numerical base they can ask you. Next thing, even you should know the point, what do you mean by linkage groups? Linkage groups will be equal to, this will be equal to what thing? Number of chromosomes in a genome, right? Now what do you mean by genome? Like I earlier said you, like every chromosome has its homologous. If I am taking 5 chromosomes, 5 together, this is 2n. If I am taking separate this and this, can I say n and n? This is one genome, this is second genome, right? So whatever chromosome is there in a genome, right? If I am taking 2n, it will be 10 chromosomes, 5 pairs. If I am separating, then it will be 5 chromosome, 5 chromosome. That 5 chromosome is indicating one genome. That 5 is indicating their linkage group. What is linkage group? All the genes on one chromosome in a genome, that you call it as one linkage group, right? Now for example, I have discussed with you Drosophila. How many chromosome it is having? 8. Now in genome H, N, I repeat, in genome N, what will be its value? 4. Now how many linkage group they must have? How many linkage groups they must have? 4. But 4 in case of the female Drosophila. Why not in case of the male, in case of the female? I must draw the sign here for the female. I repeat this point, 4 linkage group will be in a female Drosophila. But in the male Drosophila, it will be how much? It will be 5. Why so? Because in case of female, you are having 3 autosomes in a genome. Now I am speaking, I am not taking pairs. 3 will be autosome plus X and X. I will consider only X1 because that genes will be same in both. I will take 1 only. So it will be 3 plus 1 will be 4 linkage groups. Now if I am taking for the male, male is having 3 from the autosome, but if I take X and Y as a sex chromosome, these two I cannot take as 1 because both are having the genes on that will be different. So I should count it 3, 1 plus 1, it should be 5. So 5 linkage groups will be in the male Drosophila, 4 linkage groups will be in a female Drosophila. So what is linkage group? One linkage group means all the genes present in a chromosome in a genome. That's why roughly we say that it is the number of chromosome present in a genome, that is the number of the linkage group. But slightly when the chromosomal dimorphism is there, be careful about XX and XY. If XX, you have to consider 1. If XY is there, you have to consider 2 over there, right? So this is all about an important topic. Today we finished. This is based on your plus 2 NCERT book, Principles of Inheritance and Variation. What is that? Linkage and Recombination. Please do watch the video again and again if you are not getting any point clear, right? And this topic is very important regarding your NEET exam as well, right? So I hope so you have understood. Do revise it, give your best. All the best students. Take care. Bye.